So what our group tries to do is we try to pursue a precision medicine type approach. So what that means is we're trying to treat patients differently depending upon the molecular basis of their tumor type and try to match them up with the most optimal drug based upon the biology of their tumor. And so to facilitate that, about 5 years ago, we created a group called the Kravis Center for Molecular Oncology. I don't know if any of these work. And the goals of the Kravis Center are fairly simple. What we want to do is be able to define the molecular driver in every single patient who comes to see us at Sloan Kettering. So not doing this as a retrospective research protocol, but to do this in real time so we can guide patients' care. We want to use that information then to enroll patients on clinical trials if there are therapies that are investigational that are potentially better than what we could give as standard of care. And because we actually do what's called a tumor normal paired sequencing, I'll get to that in a second, we can actually identify why patients maybe got their cancer in the first place. And what we're finding is that many of the cancers that we're seeing are not completely random. Patients had a true predisposition to getting those cancers from their normal DNA. So our workhorse assay that we use right now is a type of next generation sequencing assay called MSK-IMPACT. And just very quickly to understand how this works, we try to take tumor DNA, typically from an FFPE slide. We then try to obtain normal DNA, which is not quite normal, but the most normal we can get from the blood, usually from the white cells. And right now, we can't afford to look at the entire genome in every single patient quickly. It would take too long. It would take too much money. Most patients don't have adequate tissue to do that. So what we can do is we can look specifically at the parts of the genome where we're most likely to find relevant cancer-related gene mutations by developing baits, DNA baits that are biotinylated that will hybridize to the parts of the DNA that we care about. Those baits can then be pulled down or captured using beads. We then wash off the parts of the genome that we're uninterested in and then sequence the parts of the genome that we care about and then use informatics to call what type of mutations are found in an individual patient's tumor. Really, I think the thing that's been most impactful about our initiative is that we do this not just in real time and report those results in the clinical chart, but we actually make all of the data we generate each night available to the entire MSKCC community. So we deposit this genomic data in a HIPAA-compliant way, meaning it doesn't have names or birth dates, but it's got identifiers that are anonymized into what's called the cBioPortal for cancer genomics. And everybody at MSK has access to this data as long as you have a MSKCC email address. So whether you're Craig Thompson, our CEO, or the medical student who started in the laboratory yesterday, you can access this data, you can search this data, you can look for trends or novel mutations that you might be interested in biologically characterizing or studying in patients. And you can see here, it might not be easy to see from the back, but as of a few weeks ago, we had done over 30,000 tumors from almost 28,000 patients. And this is growing into the largest clinical genomic data set in the world, at least in my opinion. People are now using this data set in ways that we didn't even predict. So I think sharing the data leads to people having ideas and making discoveries that we didn't predict a priority. So this is a report that we recently published in Cancer Cell, where we took the first 2,000 patients who had breast cancer as part of this prospective sequencing initiative. We collected detailed data on their treatment histories and whether they responded to drugs and when the tumors were collected, and then asked a very simple question. If we compare the tumors that were collected pre-treatment to those that were collected after patients had progressed or failed on hormonal therapy, did we see any difference in the genomic profile of those two different groups? And what you find is that the gray bars are untreated, the black bars are post-hormonal therapy treated patients. We see certain mutations, ESR1 being most common, that was already known, but other genes being more commonly mutated at the time of resistance than pretreatment. And these turn out to be, in many cases, acquired resistance mutations that are driving that resistant phenotype and also potential targets for therapy that could prevent or delay the emergence of drug resistance. And what this shows over here is that these resistant mechanisms are essentially mutually exclusive. If you have one, you don't have the others. So they're either present in different cells in the same patient or in different patients. This is what the cBioPortal looks like if you zoom down on a single patient And the other thing we're trying to use this information for is to enroll patients onto clinical trials. And so this is a list of mutations that this individual has. Here's the treatments the patient has seen before. And I just want to draw your attention to this mutation in AKT E17K. This is a mutation that we know now biologically in the lab drives tumor progression, but for which there is no FDA-approved drug. But we can now use this genetic information prospectively to enroll patients onto clinical trials. Here's just one example of that. This is a woman with an ER-positive tumor, breast cancer, that has an E17K mutation, who now has metastasis of this tumor to her eye, not inside the eye, but to the conjunctiva. And so this is a drug from AstraZeneca. It's a pill. You take it once a day. This is her pre-treatment image. You can see after day 11, most of the tumor has regressed, and then by day 22, she had gone on to have a complete response. And so this is the strategy. We want to be able to profile every tumor prospectively as patients are presenting, or even as their course is progressing. We want to then use that genetic data in real time to guide patients onto the most appropriate therapy, either a standard of care therapy or something that's investigational. I just want to mention a few other things that we're now trying to do with these approaches going forward. One is that one of the major hurdles in our field is that most of the mutations we identify, we don't understand their biologic or clinical significance. So one thing we do in my lab and in many other labs at the center is we try to do biology to figure that out. So I actually had a postdoc in my lab, Brooke Sylvester, whose job it was that every time we found a mutation in a particular gene called MEK1, she went ahead and made a construct of that MEK1 mutation, put it in cells, and asked whether it is an activating mutation. And so this type of functional work is really going to be critical for us to understand at the specific mutation level, not at the gene level, but at the specific allele level, what the biology and clinical importance of individual mutations represent. We're also trying to gain this information computationally. So we're trying to develop computer methods, in silico methods, where we can model these mutations and determine, without doing all that experimental work in the wet lab, whether these mutations are relevant. And I would highlight that the most predictive factor of whether a mutation is real is whether it's a hotspot or recurrent within the population. So if something is showing up again and again as a mutation in multiple different patients, that's likely not to be a coincidental finding. That's likely to be something that's being selected for because it's biologically active. And we're now using this recurrence to actually decide whether certain patients are going on to clinical trials. So this is just two examples of patients who had hotspot mutations, in this case in HER2, in this case in BRAF, that we had never biologically characterized in the lab, but just simply the fact that they were recurrent mutations drove us to test those mutations in terms of drugs that might target them. And in both of these cases, these patients went on to have good clinical responses. One of the other things we're trying to do moving forward is just expand the panel, add new genes. But what I'm most excited about the next version of our panel is that we're actually going to add probes for viral DNA. Because what we know is that many of the tumors that we see are actually driven by viral insertion, and that when these viral DNAs are inserted, they're sometimes presented as antigens and could be good patient and could represent good patients for an immunotherapy approach. And here's just one example of that. This is a patient with gastric cancer who's now had a 4-year and ongoing complete response to immunotherapy. And this was a notable tumor in that the patient didn't otherwise have the type of tumor we would expect to respond to immunotherapy. It had very few mutations, but it turned out that this was the only tumor in the cohort that was positive for EBV, and EBV is a subtype of gastric cancer that we find. And so by adding probes against the viral DNA, we're going to get essentially this viral DNA typing information for free, and we'll be able to use that information to find other patients with EBV-driven tumors that might be candidates for immunotherapy. I'll be the first to admit that not every patient can we solve the case with a targeted panel approach. This is just one example of that. This is a patient who got MSK-IMPACT. They had 11 mutations. Just take my word for it that except for the TERP promoter, these are just benign passenger mutations. I don't know what happened to it on the slide, but we then did whole exome sequencing on this patient. I guess I'll click again. And the patient had 392 mutations. Again, take my word for it. These were essentially all passenger mutations. But it turned out what we were missing in this case was an ETV6 NTRK fusion This is a functional fusion, but this was the first time we'd ever seen this fusion in a melanoma patient. Fortunately, we had a clinical trial of a TRK inhibitor. Here's this patient with melanoma. You can see the tumors all along her leg. She's gone on to this, received this drug called larotrectinib, and now has been a 2-year and ongoing complete response. So again, what is the correct amount of sequencing that needs to be done? The correct amount is enough to be able to solve the case, figure out what the key drivers are in that individual so you can match them up with the correct therapy. And just my final point, one of the limitations we've had is that we've had to get tumor biopsies to be able to do this analysis. There's now new technology which allows us to detect many of these cancer mutations in DNA that's shed from the tumor and circulated in the plasma. And we have an assay that we hope to launch in the next few months, which is using this technology, which has to be dramatically more sensitive than the type of approach we use for tumors, because the amount of tumor-derived DNA is very tiny. But this is going to allow us to expand out for this type of sequencing for patients where we can't get good access to tissue, and will also allow us to monitor changes in these mutations over time as the patients are treated, to identify new mutations that are emerging that might be resistance mutations and targets for therapy. So I'll just, since I'm out of time, just leave it with the conclusions here on the slide. I'm happy to post to my Twitter account if people want a copy of these slides that they can use, and maybe we're going to end questions after the second talk. Thanks.
Future of Care is New York City's premier community for healthcare innovation. To learn more, subscribe to our channel and visit our website at futureofcare.nyc.